I'm from biology, from genetics, and I would like to show you what we are trying to do in order to understand the language of God, if I can say, the language of the DNA. If I can have this. Okay, sorry. So, the language of life, as you may all know, we are talking about the DNA, the double-strand DNA, the double helix, discovered 60 years ago. We are celebrating at this time of the year the 60th birthday of discovering the DNA. And importantly, the DNA is telling us a story using a language, like any other language, which is built on letters, in this case only 4 letters, that makes words of 3 letters, each word only. And the words generate a sentence, which is the gene. So the 4 letters are, as you may know, T, A, C, and G. And the sentence built by letters and words is the gene. A gene for eye color is a piece of DNA with an encoded information to generate green eyes, for example. And the blue eyes is the same information in another individual with a different letters, maybe only one letter that might be different, and another eye color will be encoded. So here is the DNA, the genes, and they are packed into the cell as chromosomes. So I think that this is all what we need to know in order to understand what I'm aiming for. Several years ago, several companies, biotech companies, including one in Israel, generated a way that we can stain each strand of DNA with another color. You may know that we get 23 such strands from our father and 23 from our mother, so we have 23 pairs, and we can stain each of them by another color and look at them under the microscope, a regular microscope, not with a very, very sophisticated technology. And what I will concentrate is on what happens in cancer, one of the major problems in human health. And I'll show you that cancer is a problem of the genetic code. So what kind of problems, as I hinted already, can be found in the DNA? So as can be seen, we might have substitution of even one letter from A to G, then we'll have another information, or no information, what we call nonsense. We might have an insertion of extra letters, deletion of letters, etc. So one hallmark of cancer is changes in the DNA. They might be with the result of breaks in the DNA, so the information is disconnected or reconnected in a bad way, in a perturbed way, and it can be due to all these changes that I just have shown you. So through life, in one of the cells, there is a change that starts the cancer process and leads to many more changes in the genome. So my lab, I will quickly show you some, I think, exciting results from my research group that was already published a few months ago, and then I'll show you what I see is the revolution and maybe what we can expect from such a research. So in my lab, we asked what is the basis for this breakage and instability and changes in the DNA, and we wanted to look at the very, very early stages. And we decided to look, this is the student who did the work, at cervical cancer, which is a very common cancer in women, as you know, and as we all know, it is caused by a virus, human papilloma virus. We chose this system since, in contrast to other cancers, in this case, we know the starting point, when cancer is started by the infection with the virus, and we know within the virus who are the genes that start this process, okay? So, using a very, very nice technology invented in Paris by a scientist called Aaron Ben-Simon, that was in Israel for a few years, but now he is in Paris. He invented, together with his brother, who is a physicist, now I can connect to you maybe, David Ben-Simon, a way that you can stretch the DNA on a surface, on a small surface, and actually each strand you see on your left, stained in green, is a strand of DNA. And we can look, I will not go into the details, on the right we can follow how the DNA is replicated and how cells are propagated, but I won't go into this. And what I want to tell is that we have noticed that once we express the genes, the cancer genes that are encoded in human papillomavirus, what we generate is cells start to proliferate. Cancer is a disease that cells start to proliferate. From one cell we get two, from two we get four, etc. However, there is not enough building blocks of DNA. There is not enough biosynthesis of A, G, C, and T in the cells. This was a big surprise for everyone, including us that discovered it, because it's counterintuitive. We thought that once cells are starting to proliferate because of the cancer instructions, then the DNA will robustically be replicated. But we found is that something is wrong, and there is not enough synthesis of A, G, C, and T. And because of that, we get all these changes, all these breaks that lead to cancer. What we were able to do in the lab, is to correct it in a model in the lab, in the tissue culture. We infected cells with the virus or with the genes of the virus and supplemented the cells with extra building blocks of the DNA, A, G, C, and T. And all of a sudden, the cells continue to proliferate, but no breaks, no changes, and they lost their tumorigenic potential. So it's a simple answer to a very complicated question. We don't have enough building blocks of DNA. So, just to summarize, you can see in the upper panel, I hope you can see in the upper panel, a scheme of a normal proliferation, a normal signal for a cell to proliferate. Usually, this signal comes from outside the cell by a growth factor, which normally is regulating all the processes required for normal proliferation. However, if a cancer gene is starting the program, this cancer gene does not take care on many, many processes, including the synthesis of the building blocks. And that's one of the very early problems we have in cancer. So, about almost 8 years ago, the genome project, Human Genome Project that you may have heard about, was finished. There was a big project, an international project led by labs in the States, in Europe, and in Japan, that aimed to read the entire content of the DNA of an individual. And it was one individual. You may know it was Watson, from Watson and Crick, those that discovered the DNA, the double helix. So the DNA of Watson was actually read. So we know from chromosome 1, letter number 1, to chromosome 22, and the X and the Y, we know what is written there, the order of the letters. The cost of it was 3 times 10 to the 9 There was no technology, no computer power at that time when the project started, and all this was invented. And now we have a much, much progressed technology, and we can read the genome of all of us now for the cost of $5,000, and in 2 years it will be only $1,000. And in connection to cancer research, what I would like to show you at the last part of my talk is that now we can actually read the entire language or the entire DNA in a cancer cell. Okay? Okay. So there is a new project which is called the Cancer Genome Atlas, which is aiming to read the DNA content in many cancers. And without going into too many details, there are already 2 very important studies published in Nature just last year showing that we can actually look at different patients, at cancer cells from different patients that are morphologically and by pathologists are defined as the same disease. By looking at the DNA, we can subdivide them into different diseases, and very importantly, we can divide them by the changes in the gene and predict who is going to respond to a specific therapy and who is not going to respond, and maybe even be affected badly, negatively, by an anti-cancer regime. So I think for the future, we will be able, it's something that I can see, I can vision with some hope, okay, that we will be able to get a signature of specific cancers and based signature of the changes in the DNA that are typical for different cancers, and based on this, develop drugs that will be specific for a specific signature. So I will end by this.